Hello everyone, we hope you are so well today. My name is Paula Andrea Arroyave Ramirez and my classmate name is Maria Camila Arvachan Bargui and we are medicine students. We want to explain you all this article about the rapid identification of S. agalactiae from vaginal and rectal swabs.
Streptococcus agalactiae is a gram-positive group B streptococcus associated with the symptomatic colonization of the urogenital and gastrointestinal tracts. It is part of the usual bacterial flora of the gastrointestinal tract in the healthy people, from where it can occasionally colonize the urinary and genital tract in a persistent, transitory, or intermittent manner. In adults, it causes fever, tremors, nausea, cough, and shortness of breath. But in neonates, it worsens, causing sepsis, pneumonia, and meningitis.
The main reason for sampling of these locations is because the highest concentration site is directly connected to the digestive tract. 200 samples were collected from women in a state of gestation, older than 24 weeks without prior use within the last 30 days of antibiotics. Use a single dry swab, inserting it to the vaginal anthracis and then into the anus. Place in a standard bacterial transport medium, e.g. Amis or Swart. Label the sample so that it's clear that it's for GBS screening in pregnancy. Swabs can be self-colored by the woman herself.
The object is to develop a visual MCDA assay of the Streptococcus agalactiae targeting PCSB gene, which can achieve sensitive, rapid, and specific detection of the target pathogen.
La extracción de ADN es un proceso que se hace para recolectar material genético, para su posterior análisis molecular y forense. Y en el artículo se hizo para compararlo con otros ADNs que no fueran de la misma bacteria. La sensibilidad es la capacidad de la prueba para detectar enfermedades y la especificidad es la probabilidad de que la prueba salga negativa si no hay enfermedad. Entonces en el estudio se hizo para medir el grado de seguridad del método utilizado, ya que en otros estudios las pruebas de especificidad y sensibilidad eran muy bajas y se podían confundir con falsos negativos y falsos positivos.
Cell culture is a well-established research tool in molecular biology. It is a set of techniques that allow the maintenance of cells in vitro, preserving their physiological, biochemical, and genetic properties to the maximum. The culture was used to identify the different colonies of the Streptococcus agalactiae bacteria, which in this case took a proper stain, demonstrating the sensitivity of the test. PCR is a test used to enzymatically amplify a DNA sequence that can help establish a diagnosis. In the vaginal and rectal samples of the Streptococcus agalactiae, a specific gene was identified using this method.
En este resultado vemos la línea M, que es el valor de referencia del peso molecular. En cuanto a las líneas 1, 2 y 3, vemos que no hay mayor diferencia en cuanto al peso molecular de las bandas, pero en la línea 2 vemos que hay mayor concentración del gen PCSB y por esto se ve un poco más fluorescente que los demás. En este resultado podemos ver en la electroforesis que la banda 1 señalada con la flecha roja fue el control positivo para la galactiae, mientras que en las otras bandas no se evidenciaba nada. Por tanto, la muestra señalada con la flecha amarilla es la única que se teñió con verde de malaquita.
In the image with the red arrow, it can be seen the samples 1 to 5, how the concentration remains stable, confirming it with the electrophoresis. On the contrary, in the image marked with the yellow arrow, the concentrations are decreasing from 1 to 6 and disappearing on the 7th, which is also confirmed with electrophoresis. The Coccus agalactiae identified with malachite green, while the other 21 samples did not obtain this staining. We can also verify it in the electrophoresis indicated with a blue arrow, seeing that for example 1 to 7 the concentration of bacterium was shown, while in the other samples nothing is observed.
As we can see, we have three arguments. The Carrillo's argument isn't agreed with this article, and the arguments of Phillips, Elbihiri, Wong, and Lee are agreed with this article.
Conclusions. Molecular biology tests have guaranteed early diagnosis, which can lead to effective and immediate treatment to obtain good results. This science allows the deep study of microorganisms that can be pathogenic and deadly, even before someone is born, for which biology provides a preventive way to be prepared. On the daily basis, molecular biology has become very important in clinical practices because it provides alternative routes for the treatment of pathologies that can save a life with only the early detection of this, as is the example of this study. Thank you!